So, hello guys, what's up? I hope you are doing well. In this particular video, we will discuss about the binary fission in Paramicium or asexual reproduction in Paramicium. So that's the video number 5 on Paramicium. Before this video, I have uploaded 4 more videos on Paramicium where I have completed the nutrition, systemic position and morphology, osmoregulation and locomotion in Paramicium. So if you want to see that particular videos, you could check the description of that particular video to get their links. So in this particular video, we will discuss about asexual reproduction in Paramicium. So as we know, that's the structure of Paramicium. As we know, Paramicium is a protozoan and it could perform both sexual as well as asexual reproduction. Okay, in previous video, we have discussed about micronucleus and macronucleus and micronucleus of Paramicium. So before starting the reproduction in Paramicium, we have to know about these two structures. The first structure is known as, the large structure that you can see is known as macronucleus, and the smaller one that you can see is known as micronucleus. Okay, these structures are very important. Okay, without these structure, it could reproduce. Paramicium couldn't reproduce. Okay, so first one is macronucleus. So what's the function of macronucleus? There are two functions of this structure. Means macronucleus perform two functions. Its first function is to control the metabolic activities of that entire entity. Means it controls the metabolic activities of Paramicium. Its second function is to perform asexual reproduction. First function is to control the metabolic activities, and second function is it helps in asexual reproduction. The second body that you can see is known as micronucleus. This micronucleus help in asexual reproduction. As we know, in Paramicium, both type of reproduction could occur. Okay, it could perform sexual as well as asexual reproduction. So if this Paramicium performs sexual reproduction, it is performed by micronucleus. It would be performed by a micronucleus. Or if asexual reproduction occur in amoeba, it would be carried by macronucleus. Okay, it helps in asexual reproduction and it helps in sexual reproduction. Sexual reproduction will be discussed in a next video. In this particular video, we will discuss about asexual reproduction in Paramicium. Okay, so let's start. Now, if Paramicium have to reproduce asexually, then it is performed by a special method known as binary fission. Okay, it is performed by binary fission. Now, what is binary fission? Binary fission is a mode of asexual reproduction in which an organism, a complete unicellular organism, split in two individuals. Okay, a process in which a complete unicellular individual divide in two entities or organisms is known as binary fission. The binary fission may be of 3 types. First one is transverse binary fission, second one is longitudinal binary fission, and third one is irregular binary fission. Okay, in amoeba and euglena, we have already discussed the irregular binary fission and longitudinal binary fission. Today we will discuss the transverse binary fission. Okay, so let's start the transverse binary fission. Okay, what are our objective in this particular video? We have to split that Paramicium in 2 entities, okay, through transverse binary fission. Okay, let's start. Now that's the body of Paramicium. Before starting the reproduction, I have to tell you about some other entities. Okay, these are discussed. Now here you can see 2 structures. What are these 2 structures? These are the contractile vacuole complexes. Okay, that structure is a contractile vacuole complex and that structure is a contractile vacuole complex. A normal Paramicium's body contain 2 contractile vacuole complexes. Okay, one at anterior end and one at the posterior end. Okay, in the contractile vacuole complex, here you can see the dots two red color dots, you can see these red color dots are known as contractile vacuole. This entire entity is contractile vacuole complex, and the red circle that you can see is the contractile vacuole. Okay, so let's start. Now that's the first stage. In first stage we done nothing, just draw the diagram. Okay, now in second step, now we have started. Okay, we have started. Now in second step, what we can see here, we can see in first structure we can see the paramecium has a slipper shaped body. Okay, in second step its body get distorted. Okay, somewhat distorted, means in first case it has a slipper shaped body, but in second stage its body got rounded. Okay, its body got rounded. Here you can see in that structure that you can see it has a slipper shaped body, and in that particular structure its body got rounded. Okay, that's the first point in second stage. Okay, in second stage. And what's the other change in second stage? First one is body get rounded, and second is its cilia increases in number. Okay, its cilia increases in number. In second stage, paramecium increases their cilia, number of cilia. Okay, here you can see the cilia get more dense. Okay, as compared to the first one, the cilia of first one. Okay, so here you can see the points. What happened in second 2 points I have already told you. Okay, now what are the next points? Here you can see pore for contractile vacuoles are formed. Okay, these are the old contractile vacuoles. Now we have to split the complete organism in two parts. Now we require two more contractile vacuoles. Okay, because we have to make two bodies. Okay, one body contains two contractile vacuoles. So how many bodies these two require? These require 4. Okay, so we have to make two new contractile vacuoles. Okay, so how they form? Here we can see pore for contractile vacuoles are formed. At first, two small pores are formed at the back side of macronucleus. Here we can see two pores are formed. Okay, so that's the second point. That's the second stage. Now what happened next? Here we can see second cytopharynx start to form. Okay, now here we can see that particular structure is known as cytopharynx. Okay, now we have to divide that particular body in two parts. Okay, so first we have seen two rounded bodies appear. Okay, that would be converted in contractile vacuole. Okay, and other is, here we can see the cytopharynx. Now in second stage we can see a new cytopharynx starts to form. Here we can see that's the old cytopharynx. Now a new cytopharynx starts to form. Here we can see. Okay, here we can see now new cytopharynx starts to form. Okay, so that's the second stage. Now what happened in third stage? Here we can see what happened in third stage. Now third stage is the most important stage. Okay, now that particular structure that we can see, okay, these starts to move at anterior and posterior ends. Okay, and also these pores changes into vacuoles, and these vacuoles trying to increase their size. Okay, their size now increasing continuously. Okay, so in next step we can see, here we can see now that small bodies change into that particular body, and that small body change into that particular body. Now we can see, here we can see it is increased in size. Here we can see, okay. Now what happened in third stage? Here we can see some constriction appear. Now I have told you we have to divide that particular organism in two sets. Okay, now I have told you already, third step is most important step. Okay, now at first we can see a constriction appear at the unicellular body. Here we can see, means in such a way, okay, that in such a way. Here we can see two constriction appear Okay, they form two constriction. Now also we can see here, we can see the points. What happened in first point? We can see constriction appear, that's the constriction. Okay, now second point where that we can see closed mitosis in micronucleus. Now the micronucleus, here we can see the micronucleus, closed mitosis starts to occur here in that micronucleus. Closed mitosis. Closed mitosis is a special type of mitosis, and I will make a separate video on that particular topic. Okay, so closed mitosis starts in micronucleus, and that's the second point. And next one is amitosis is in macronucleus. Okay, now that's the macronucleus. It starts, that's the micronucleus. In micronucleus, closed mitosis begins. Okay, and in macronucleus, amitosis begins. Now what is amitosis? Amitosis is the simplest mitosis in which the body or the cell completely divides in two sets, but there is no spindle formation. Okay, here you can see that's the macronucleus. Okay, here we can see that's the macronucleus. In amitosis, what happened? The particular structure stretches and simply divides into. Here you can see the macronucleus. What happened? It stretches. Okay, it stretches. There is no spindle formation in amitosis. Okay, simply what happened? That particular body stretches. Here we can see that particular body stretches, and after some time there appear one constriction in such a way. Okay, in next step we would see it. We would see that now. So and I have told you this body trying to move upward and this body trying to move downward. Now this body came here and this body came here. Okay, now you can see that particular portion that you can see, it would make the upper paramecium, and that particular body that you can see makes the lower, would make the lower paramecium. Okay, now in second step, what would see? Simple constriction appear. Here simple constriction appear and two bodies of paramecium we would find. In that particular structure we can see what happened. A constriction appear here, and at the site of constriction a septa is formed, means a layer is formed in such a way a layer is formed, and that particular bodies we would find. Okay, so these are the daughter paramecium. Okay, so that's the asexual reproduction in paramecium. In next video we will discuss the sexual reproduction in paramecium. So I hope you found this video useful. Thank you.